Do you know that our body needs glucose, which comes from carbohydrates to survive? So the brain can only use energy that has come from glucose, yet the body can only store enough glucose to last for less than 2 hours. So what happens if you only eat meat for a day? What does the body do to prevent us from dying? To answer this question, it's through gluconeogenesis, which is pathway used by the body to create glucose from other molecules. This is the pathway that prevents us from dying when we have not had any bread, vegetable or other carbohydrate for a few hours. The metabolic pathway, gluconeogenesis, is the synthesis of glucose from 3 and 4 carbon molecules, such as pyruvate. These molecules can be synthesized from some amino acid into triglycerides. We will also use gluconeogenesis if too much of the glucose has been converted into pyruvate through glucolysis. The process can also be reversed and turning back the pyruvate into glucose for the storage and also usage for any part of in our body.
Step 1. Pyruvate convert into phosphoenolpyruvate. This is the step that differs from glycolysis. Pyruvate will be processed by pyruvate carboxylase into oxaloacetate. This enzyme within the mitochondria requires ATP for its function. Because oxaloacetate cannot pass through the mitochondria membranes, it must be converted into malate by malate dehydrogenase. Malate can then cross the mitochondria membrane into the cytoplasm by malate aspartate shuttle, where it is then converted back into oxaloacetate with another malate dehydrogenase. And oxaloacetate is converted into phosphoenolpyruvate PEP via PEP carboxygenase. This process releases carbon dioxide molecule and the enzyme requires GTP for its function. At last, phosphoenolpyruvate is formed. The formation of phosphoenolpyruvate, the steps are similar to glycolysis but in reverse. Step 2. Phosphoenolpyruvate rearranged into 2-phosphoglycerate. Step 3. 2-phosphoglycerate rearranged into 3-phosphoglycerate. Step 4. 3-phosphoglycerate gets another phosphate added, forming 1,3-bisphosphoglycerate. Step 5. 1,3-bisphosphoglycerate rearranged into glyceraldehyde. Step 6. Glyceraldehyde combined with another 1,3-bisphosphoglycerate to form fructose 1,6-bisphosphate. Step 7. Fructose 1,6-bisphosphate lost a phosphate and become fructose 6-phosphate by enzyme fructose 1,6-phosphatase. Step 8. Fructose 6-phosphate rearranged to form glucose 6-phosphate. And the last step, which is step 9, glucose 6-phosphate lost the phosphate to form the final product, which is glucose by enzyme glucose 6-phosphatase.
As stated before, gluconeogenesis occurs in liver and kidney. But during fasting, kidney is the major organ to produce glucose. Important substrates for gluconeogenesis are lactate, pyruvate, cholesterol, and amino acid. All amino acids except leucine and lysine are glucogenic amino acids. Alanine is the major glucogenic amino acid. These amino acids are converted to citric acid cycle intermediates, then to malate and to glucose. In humans, it is not really possible to convert fatty acids into glucose. Now we have learned the required substrate for gluconeogenesis. We will continue by explaining the sequence of the reactions that make up this pathway. So, as we have learned before, Glycolysis, the breakdown of glucose, consists of 10 reactions, 7 of which are reversible and 3 of which are irreversible. So, now because of these 3 irreversible reactions, which are step 1, 3 and 10, gluconeogenesis does not simply occur by the reversal of glycolysis. This reaction must be replaced by reactions that favor the production of glucose. Since we are doing the reversal of glycolysis, the first obstacle is reaction 10 of glycolysis, the irreversible of phosphoenolpyruvate to pyruvate by pyruvate kinase. To yield phosphoenolpyruvate for gluconeogenesis, we need two enzymes, pyruvate carboxylase and phosphoenolcarboxy kinase. The reaction goes as such: pyruvate carboxylase converts pyruvate to oxaloacetate in the mitochondria. Oxaloacetate is converted to malate or aspartate, which travels to the cytosol and is reconverted to oxaloacetate. And then phosphoenolpyruvate carboxy kinase converts oxaloacetate to phosphoenolpyruvate. Phosphoenolpyruvate then go opposite direction.
Gluconeogenesis is a pathway used by the body to create glucose from other molecules and an important pathway that allows the body to store needed energy for the brain in the form of glucose. It is essentially glycolysis, which is the process of converting glucose into energy in reverse. There are 9 steps in the gluconeogenesis process. However, a few steps are not energy favorable for reversal to different enzymes, which is step 7 and step 9, and added steps are included in order to overcome the energy differential. Pyruvate can be turned directly back into glucose, and some amino acids also can be used as a glucose store. In addition, protein can be used to create glucose storage. Ultimately, glucose is extremely important because it is the only energy source that our brains can use to survive.